नवयुग शिक्षण प्रसारक मंडळ चापोली संजीवनी महाविद्यालय चापोली welcome you in e-learning platform prepared by myself Dr. Shubhangi Chavan, Assistant Professor and Head, Department of Botany. Now, Swami Ramanand Tirth Marathwada University Nanded Semester Pattern Curriculum under Choice Based Credit System CBCS for undergraduate program, Faculty of Science and Technology, subject Botany, year BSc second year. Now student, last period C, permanent tissue, Sclerenchyma. Now the start next tissue, Sclerenchyma. Sclerenchyma composed of thick walled cell, often lignified parenchyma and procaryma. The main function is the provide mechanical support. They show great variation in shape, size, organ and development. They are also act as supporting tissue which results from starching and binding of plant organs without any damage to the thin-walled suffered cells. At maturity, Sclerenchyma cells do not possess living protoplasts. Now, the cell wall are strong and uniformly thickened, called sclerotic parenchyma. Sclerenchyma is further divided into fiber and scleroids. First of all, fibers. Fibers are elongated, narrow, thick, with pointed or blunt end. The cell wall of fibers are lignified. They are present common in vascular tissue but also occur in the ground tissue. They occur in bands or form unitripleted hollow cylinder. The pith of fibers are small, round or slit-like, present on the wall, few in number and many. The middle lamina is large and coniferous. One maturation of the cell, the protoplast disappears and permanent cell becomes dead and empty. They have great tensile strength, flexible and elasticity, which enable plant organs to withstand various strains caused by outer agents. In TS of fibers appears rounded or polygonal in shape with a well-developed lamina. Sometimes lamina become very narrow due to excessive wall thickening. One of the bases of their portion in the plant body, fibers are divided into xylem fiber and extraxillary fiber. Now, first of all, xylem fiber. Xylem fiber are also known as wood fiber. These are associated with the xylem. Xylem fibers are developed from the same meristematic tissue which gives rise to the other xylem elements. These fibers helps in conduction of water along with mechanical support. These cells possess lignified secondary walls. They vary in size, shape, thickness and wall. They process various pits. Xylem fibers with the very thick secondary walls and simple pits are called labriform fibers, while thin-walled are usually broad red pit fiber tracheids. Both the xylem fibers are scripted secondary xylem, and certain dicotyledons plant has gelatinous or mucilaginous fiber. Xylem fibers have more alpha cellulose than lignin and have great capacity of water absorption. Now, extracellular fiber. Extracellular fibers are related to the phloem, occurs in various tissue outside the xylem such as cortex, pericycle and phloem. The fiber form continuous cylinders in cortile fibers present in the peripheral region of the vascular cylinder very close to the phloem are known as pericycle fiber. Extracellular fibers are also called best fiber, which are classified as follows. Now there are three types: phloem fiber, cortical fiber, perivascular fiber. First of all, phloem fibers. Fibers originating in primary or secondary phloem. Secondly, cortical fiber. Fibers originating in the cortex. And last, perivascular fiber. Fibers found in the peripheral region of the vascular cylinder but not originating from the phloem. Now, the diagrammatic representation of Sclerenchyma tissue. First figure Sclerenchyma TS and LS. First of all, in transverse section of Sclerenchyma and in longitudinal plane of Sclerenchyma. And next figure, type of fibers. A type of fibers, the diagrammatic representation of type of fibers. First of all, A, labriform fibers, B, fiber tracheids, C, septate fibers tracheids, D and E, gelatinous mucilaginous fiber in longitudinal plane and in transverse section respectively, and F and G, unseptate and septate phloem based fiber respectively, the type of fibers. Now, extracellular fibers are long spindle shaped with tapering ends. They are usually unbranched, but branched fibers are rare both in monocotyledons and dicotyledons. Plant fibers are of great economic importance. There are more than 44 families which are great source of commercial fibers. They include flax, jute, hemp, ramin, sisal and roasting hemp. Next, sclerites. Sclerites are widely distributed in the plant body. Scleroids are short or irregular. They are also known as stone cells or sclerotic cells. They are broad, occur single or in groups. They are isodermistic but may be spherical, oval, stellate, T-shaped or cylindrical in shape. They are dead cells with small lumen, but the protoplast of some scleroid remain viable for long period of time. They are found in the cortex and pith of gymnosperms, and dicotyledons arranged singly of group. In leaves, the mesophyll is completely perimetered by scleroid. Scleroids are also common in fruit and seeds. In fruits, they are discussed in the pulp. Due to scleroid only seed coat in hard and strong. The secondary wall of the scleroids are typically longified and vary in thickness. Numerous simple pits are present and their cavities are less branching canals. They are also associated with xylem and phloem and parenchymatous tissue. Example, pith and cortex of stem and petiole. Roots, leaf, mesophyll and flesh fruits. Scleroids are distributed randomly or they occur in specific position such as end of venlet called terminal scleroids or near the margin of leaves called margins scleroids. One of the basis of their shape and thickness of wall scleroids are classified as, first of all, astroscleroids. They are star-shaped scleroids. These scleroids are with lobes projecting like hairs, are commonly found in the intercellular space of the leaves and stems of hydrophytes. Example, nymphia. Next, second, B. Brachyschoroids or stone cells. These stone cells are short, roughly isodermistic in shape. They occur in the cortex, phloem and pith of stem and in the pulp of fruits. Example, Conimonia. Third, microscleroids. They are elongated, columnar, more or less rod-like cells forming placids like epidermal layer of many seeds and fruits are found in xerophytic leaves and stem cortices. Example, leguminous seeds. Now, the diagrammatic representation of Sclerenchyma tissue. First figure, scleroid showing pits in surface and side view. And next, type of scleroids. First of all, A. brachyscleroids. B. Astroscleroids. C and D. Microscleroids. And E. Osteoscleroids. And F. Trichoscleroids. The diagrammatic representation of type of scleroids. Now, D. Osteoscleroids. They are bone-shaped or barrel-shaped scleroids and dilated at ends. Such scleroids are commonly found in the hypodermal layers of many seeds and fruits. They are also found xerophytic leaves. Example, many monocotyledons. And last, trichoscleroids. They are hair-like branched and occur in the intercellular space in the leaves and stems of certain hydrophytes in the earlier root of monstera and the leaves of olive.